And thank you for the introduction, and thank you for inviting us to be part of this amazing event, because we really want to spread the word about the lymphatic system. This is just a tiny part of the team. We got people in the hospital that is working on helping patients with this condition, and there's a great team in the research department in the university that also works on this, and I can see a few of my colleagues up here at the front. So there will be a lot of people to ask questions to afterwards. The 4 people on the screen here are the 4 people that are going to give you a little taste of what the lymphatic system is and what happens when it goes wrong. We got Professor Zaha Mansour, Professor Peter Mortimer, and Dr. Christiana Gordon, and they're all clinicians and working in the hospital. And then you'll hear from myself as well, and I'll be representing the research that we're doing here. So these are the questions we're going to cover, if Zaha doesn't sneeze her way through. So Chris will come up first and talk a little bit about what the lymphatic system is and what it looks like. And then Zaha will talk about when it's gone wrong. We get primary lymphedema and give you an idea of what patients look like with this condition. And then I will come and tell you about the studies we do here and the genetics work we're doing. And then Peter will round up with explaining how we help the patients. Okay, so Chris, will you come and continue? Yeah, if you want me to help. I don't know if you need to hold, or you know you've got a thing there as well. Do you mind? Oh, no, I want to do it this way.
It's a good job we know each other well.
Yeah.
Yeah. Thanks, Pia.
And this is forwards.
Mm-hmm. Okay. Thank you, Pia. Thank you, Julian, for the Thank you, Pia. Thank you, Julian, for the opportunity to speak tonight. So I'm going to set the scene about the lymphatic system. I'm going to hopefully inform you what the lymphatic system is, where it is, and what we think it does, and then a little bit about what happens if it doesn't work properly, and then setting the scene about why does it fail, and explaining the difference between secondary lymphedema and primary lymphedema, and then I'll hand over to Zaha. So what is the lymphatic system? Well, if you ask most doctors and nurses, medical students, they'll sort of shrug and go, not really sure. 1 or 2 might say, you know, in a slightly snarky tone, well, I've heard it's the waste disposal system or the sewer network of the body. I think that just reflects the lack of respect that the medical and research community have been showing the lymphatic system for many years, unfortunately so. But let's start off about thinking, well, where is the lymphatic system in our body? It's everywhere. Lymphatic vessels are in all of our tissues. It's this network of see-through, fragile vessels that will then connect into larger vessels, draining into our lymph nodes, which hopefully many of you have heard about, into larger vessels, and then draining into the heart. And these vessels, when they become larger in size, will have valves in them to keep everything draining in one direction, and they'll pump every 8 seconds or so to keep the fluid moving in the right direction. When we think about the drainage system of the body, most people will automatically think about the cardiovascular system, and this is a circuit that keeps oxygenated blood going around the body to the tissues. But most people won't think of the lymphatic system when you think about circulation and drainage. So the lymph system is everywhere, and it's draining the fluid from the tissues, and I'll try and explain that a little bit more. If you think of the body, if you think of the tissues, you've got your arteries carrying oxygenated blood. The heart is pumping that to the tissues, and then the tissues are taking the oxygen that it needs and then giving the deoxygenated blood back to the heart through the veins, through the venous system. Where the higher pressure in the arteries goes into the lower pressure of the veins, this happens at the capillary bed. That's that reddish-blue network you can see there. At this pressure change, you've got a lot of fluid filtering out of the blood vascular system, and this fluid's going out into our tissues, but it can't stay there or else will swell up. So it has to drain somehow, and it's the lymphatic system that's crucial in that process. So this cartoon is showing you that the green vessels, I don't know why they're always pictured as green, they're see-through really, but these lymphatic vessels are trying to suck up all that water, that interstitial fluid that's in our body tissues, and draining it away. And when it's in the lymphatic system, that fluid is now called lymphatic fluid. So, as I was trying to say, eventually the lymph system will connect to these larger lymphatic vessels, and then this fluid, this unwanted fluid, will drain back into the heart through a big vein, and eventually that unwanted fluid will get filtered round, and perhaps some of it we will urinate out.
Is that osmosis from the veins outwards?
It's a complex process to do with pressures, and osmosis is part of it, yes. But I'll defer your questions to the expert physiologist later for that one, if I may. So just another cartoon to try and explain that that box at the top there, that's meant to be our heart pumping the oxygenated blood. So the oxygen's come through from the lungs, and it's oxygenated the blood, and it's going round, and you've got this nice circuit system going round from arteries to veins, and then your lymphatic system in green is doing its best to just drain up all that fluid that's being filtered out. And you've got vessels there connecting to lymph nodes and collecting to larger vessels. So what does the lymphatic system do? Well, I've talked a lot about fluid filtration and drainage of fluid from all the tissues in the body, but that's not its only job. But unfortunately, not many people realise it has other jobs. It's vital for immune surveillance in the body. So its job is to detect when infections are trying to invade the body. And this is happening all the time to us, usually through a little cut in the surface of the skin. And when that happens, the lymph system is vital in helping your immune system recognize that a bacteria or a foreign body has come in, sends a message to your immune system. It then mounts up a response, and your immune system goes back to that cut, to that bug that's come in, and will fight it off. And you don't realize any of this is happening. You don't get an infection. What else does it do? Well, it drains other cells from our tissues. For example, inflammation cells and cancer cells. So if somebody has cancer, the cancer can spread or metastasize through our lymphatic system. So now we know what the lymph system does. What happens when it fails? When your lymph system isn't draining the fluid properly, you get lymphedema, you get swelling. And some of you may have heard of the elephantiasis word describing the skin changes. And here's a picture of somebody with very poorly controlled lymphedema who hasn't been able to access the care that they need. They've got swelling. You can see that their legs are much bigger than they should be. And they've got this cobblestoning appearance of the skin as the skin is inflamed and upset. What you might be able to appreciate is the yellowish brown discoloration there. And that's not dry skin. That's lymphatic fluid that's actually leaking through the skin and drying on the surface. And that's because the pressure is so high in those legs, the fluid is escaping through the path of least resistance. But with good treatments, we can certainly help these patients. What else can happen? Well, we know that the lymphatic system is important for immune surveillance, fighting infections. So when the lymph system fails, you get infections such as cellulitis, which is the medical term for a bacterial infection of the skin, when it becomes red and spreads and the patient feels feverish and unwell. You can also get verrucas, a viral infection, an athlete's foot, a fungal infection in these areas where the immune system isn't able to work as well as it should. So here's a patient, a lady that has lymphedema of her right leg, so you can see it's bigger than the left leg, but you can also see this red rash. This isn't an itchy rash, this is going to be a red hot rash, and she's going to feel feverish and flu-like. And she's in urgent need of antibiotics. But she will need antibiotics for longer than someone without lymphedema because it'll be harder for her to fight this infection. Sorry for the slightly gruesome picture here, but this is athlete's foot. And this is another complication of someone that has lymphedema. They often get athlete's foot. And you may think, oh, so what? It's just an itchy rash between the toes. But you can appreciate perhaps that there's some splitting of the skin here between the toes. And unfortunately, bacteria are very clever, and they might go in through those athlete's foot splits and then create a cellulitis. So the fungal infection probably will lead to a bacterial infection if the patient's not careful. So moving on to the second part of my talk, why does the lymphatic system fail? Why does somebody get lymphedema? When we see someone in clinic and they have lymphedema, they have the swelling, we try and work out which category do they fit in. Do they have secondary lymphedema or primary lymphedema? Secondary lymphedema happens when someone who is born with a normal or a healthy lymphatic system has an event where something damages their lymphatic system and it can no longer work properly. So there's a problem with the flow of the lymphatic fluid and they become swollen in the damaged area. And there's more than one cause of secondary lymphedema, and this list is long but not exhaustive. But just to raise awareness that some blood pressure medicines, amongst other medicines, can cause damage to the lymphatic vessels. They stop them from pumping in some situations. Somebody who has cancer, or more often the treatment for that cancer that might involve removal of their lymph nodes, can cause lymphedema. Repeated infections of the skin can cause lymphedema, so cellulitis that keeps happening will eventually cause lymphatic vessel damage. Somebody with very severe and chronic inflammation of the skin can be unlucky and get lymphedema. For example, somebody who has terrible hand eczema that's very poorly controlled, over time may get hand swelling because of lymphatic vessel damage. And then you have the other situations where it's easier to understand why a lymph system will fail if there's trauma or surgery or a vessel is directly cut. But a large number of patients in our clinic are coming up with swelling of the lower limbs, swelling of the legs, because of varicose veins or because of obesity. So this is a problem that we're seeing more and more in our patients in clinic. And as I said, there are other causes. But if we take a step away from secondary lymphedema for a bit and think about primary lymphedema, which will be the focus for the rest of the evening, when does this happen? Well, primary lymphedema is swelling that is caused by a developmental problem of a person's lymphatic system. It probably happens because of a genetic mistake that the person was born with. Even if that gene mistake is present when that child is born, the swelling may not show itself until later. And I don't just mean in 6 months' time, it can happen many years later. So we are learning that primary lymphedema is not just one disease, and there's a lot more that we will be learning about primary lymphedema tonight. And I will hand you over to Saha. Thank you. Well, we're going to put that one there.
Where does that go?
I don't know.
Is there 2 microphones? I feel like I'm going into space. You can certainly hear me now. I'm a geneticist. I've been doing this clinic with the team for over 20 years. And we've come a long way in learning about primary lymphedema and the causes of primary lymphedema. We used to think it was one disease. It was either primary lymphedema or secondary lymphedema. And if you go to some hospitals, that's what they still think. But actually, through being very, very careful about looking at these patients, examining them carefully, doing some more investigations, we have managed to dissect it into many, many different conditions. And each of them have a different mechanism, different cause. And we're hoping, the dream is, that by understanding the mechanism and the cause, you could treat each patient individually, personalised medicine, targeted medicine, and that's the aim of all this work. So I'm very interested as a geneticist in the phenotype, and the phenotype is the pattern of abnormalities that you see as a result of a mistake in the same gene or a damage due to the same drug. So it's how it presents, what aspects do they have, a big nose, sticking out ears, 2 extra fingers, that makes up the phenotype of these patients. So we're very, very careful about looking at the phenotype of the patients we see in clinic. And we consider very carefully when they developed the lymphedema. Was it at birth, so congenital? Or was it late onset? So as Christiana was saying, you can have primary lymphedema that presents much, much later. It's still genetic, it's still predetermined, if you like, but they don't develop the swelling till later. Or is it one leg more than the other? Is it asymmetrical? Is it localized? Is it somatic, which means that it's a problem just in that segment? So each of these conditions here, we've only picked 3, there are hundreds, well, not hundreds, but there are many. many different conditions, I've just picked 3 as an example, have a different cause and a different underlying mechanism. So the first one is a condition called Milroy Disease. It's one of the more common forms of primary lymphedema. You can see here at the bottom a father and daughter with congenital primary lymphedema of the back of the foot. When you examine them, we've got quite good at this diagnosis because it's very characteristic. It's mainly the back of the foot, the dorsum of the foot. The nails are small, upslanting, we call it Bryce sign after one of our colleagues, upslanting nails, small dysplastic nails. There are deep creases in the toes. You can see these deep creases. Oh, it works. So it's a very distinctive feature, and these are twins with Milroy disease. So you can see it's genetic. There's more than one family member. And that's all they have. They don't. Some of the boys have fluid around the testes, but otherwise they're completely normal. So you see this, you worry about all sorts of diagnoses, but if you can make this diagnosis, it's a relatively benign condition. And we found that lymphoscintigraphy can help us with the phenotyping because you can have specific changes on the lymph scan to help you make a diagnosis. So this is a normal, apparently Peter, it's normal. This is Professor Mortimer's lymphoscintigram. They injected his toe web spaces. I've refused. And these are the lovely, beautiful lymphatic tracts to the inguinal lymph nodes. So it's showing normal uptake of the lymph from the periphery to the inguinal lymph nodes. This is a patient with Milroy disease, and this is absolutely typical of Milroy. You inject the radioactive isotope in the feet, and you see nothing. There's no inguinal lymph nodes, no tract. And we thought that the lymphatics were missing, but actually when you do a skin biopsy, you can see the lymphatics, but they don't take the fluid up. So there is a mechanistic problem with the lymphatics. So this is very different to this condition. This is another autosomal dominant condition, lymphedema distichiasis. They get swelling, but I've never seen the swelling at birth. It usually presents from the age of 8, 10. We've seen 40-year-olds presenting with swelling. So it's a late onset lymphedema. It's not congenital, but it is genetic. And the lymphoscintigram, look, it's completely different. This is showing the outline of the leg. So what's happening is the radioactive isotope is going up the leg and it's coming back down. And Pia's going to explain why. Because we were instrumental in identifying this gene, and it's really important for the valve development. So you'll see that in a minute. These patients come in, they sit down, and they've had swelling of their legs for years. Nobody's known why. And my favorite time is when you say, do you have eyelashes that cause irritation and itchiness and infections in your eyes? And they say, how did you know that? And you know immediately that they have lymphedema distichiasis because it's a really rare thing to have distichiasis. And distichiasis means double row of eyelashes, but they don't have a double row. They just have these aberrant eyelashes that grow from the inner eyelid and grow onto the conjunctiva and cause irritation. It's a really good handle, a clue for this particular form of lymphedema. It's also important because this gene can cause congenital heart defects, and we've had a few patients. We now send them all for a heart scan, and one of them ended up with surgery within 6 months. So, if you know a little bit about the condition, you can look for the complications associated with it. And then this is a rare but problematic, condition. This chap is completely normal, but he has a gene defect in just some of his cells, and Pia will explain how that happens. So this is a very large overgrown leg, and this leg also has some overgrowth. If I say to you that this, on the right side, this big toe is the normal big toe, you can appreciate that he has huge overgrowth of his toes and his limbs. He also has, what you can't see very well, is vascular malformation, he has lymphatic problems, and he has venous problems. And this leg continues to grow. This is another lady. She made a documentary on the television, so I can tell you about her. You can see how slim she is, very fit, very slim, because all her calories and energy go into the growth of her legs. So she has a progressive overgrowth of the legs because of this particular gene mutation, which is in both of them. And it was such a problem that she ended up going to America for an amputation of that leg because it was heavy, she was getting recurrent infections. And then the stump grew as well, because the drive from this particular mutation it's also, you get similar mutations in cancer. There's a very strong drive for growth in this particular gene. And then by finding these genes, it can lead to better treatment. Are you talking about the treatment, which Pia will mention? So our chap who I showed you before is actually on a relatively new treatment and has had some success. Okay. So all our hard work over these 20 years have led us to this classification, which we've published a few times, and it's showing all the different causes of lymphatic problems. You can see here lymphedema distichiasis in the late onset, Milroy in the congenital, and the PIK3CA somatic mutation up here. But you can see also that there's lots of conditions I haven't even touched on today. So we're just starting to fit these pieces of jigsaw together, understand the mechanism, and our belief is that it will lead to better treatment, because when you hear Peter's talk, you'll see it's still quite limited. Thank you.
Okay.
Thank you.
Thanks a lot, Sarah. It's fantastic. So now you're getting a better idea of what the lymphatic system is and what happens when it goes wrong. And Sarah has also mentioned a little bit about some genes, and I'm going to go into more detail about that. So we now got the three phenotypes that Sarah mentioned. And we also given them a name now. So we got Milroy Disease, we got Lymphedema distichiasis, and we got PROS. And these were the phenotypes that represented each of these groupings. So what we can do now to understand this better and what's going on in these patients is to look at the DNA and analyze that and find out if we can find a genetic defect in these patients. But first of all, for those of you who are maybe not so familiar with what DNA is, I've just put in a few slides so that we all can understand what's going on here. So if you think about your body and all the cells that you have in your body, we have trillions of cells constituting the human body. And in each of these cells, so this is a cell, we have something called a nucleus. So this is the core of the cell. This is where the most important part of the cell is. And inside the cell, we got small units called chromosomes. And if you look at those in detail, you will see that they consist of this DNA material, which is just a long molecule that is sort of twisted up, and it's sort of compacted into these units, storage units. So the chromosomes are just storage units for DNA. And just a fun fact, because this is really incredible to think about, because you can't really see cells when you look at your body. You don't see them because they're so small. But if you think about a grain of sand, which all of you will know about because you've probably been to the beach, or you might have a timer at home in your shower telling you when you should get out of the shower, which is also a little sand thing. If you imagine each cell is a grain of sand, your hand, just the cells in your hand, would be the size of a bus. So that's just to give you an idea of perspective here, what we're talking about. So the DNA, which is inside the nucleus, inside this grain, is even smaller, but it's still a very important part of our body. And we can now read DNA. And when I say read, of course, it sounds amazing when it's so tiny, but we have found a way of how you can actually read the DNA, and that's called the genome. And there was a human genome project that was running for 13 years, and they published the human genome. It was a very expensive project. It cost over £2 billion, and it took a massive team of people to read this human genome, and they just did one person, just one human genome. So why all this effort? Why all this work? Why all this money? Why was this project so useful? Well, it was useful because it gave us this reference sequence where we can now read the DNA, as I said, so we now know the sequence of these molecules in the DNA strand. And when we then have patients where we know that there's a problem, we can then take their DNA and match it up or align it up to the reference. And then when they don't match, that's where we have an error in the genetic code, and that can then help us to identify the problem. So this is what genetic studies is about. It's taking the reference genome that we have, matching patients up to that, and try to identify where we have errors. So I've taken the code that we can all read, because the DNA code is only written with 4 letters. They've been assigned with 4 letters, but I thought if I made a sentence that makes sense to everybody here, we can maybe better understand what I'm talking about. So when we then have problems in the code, like small changes, and often these genetic conditions that we see is only a single letter that has been changed in this sentence, and then it starts creating havoc. But sometimes the change doesn't matter so much, because maybe you could actually imagine that the fit cat could better eat a rat because it would be more fit to actually catch the rat than the fat one. And also maybe the fat cat would have to eat hats because it's not running very well, so it can't catch the rat. But it will still make sense. But you could also have situations where maybe you have lost a letter in your code, or you have inserted a letter in your code, because these things can happen as well. And then you can see the sentence stops making sense. And then that is really disturbing for your cells, because they're reading this DNA all the time. And if there's any errors in the text, just like yourself, the body will go, ooh, I can't cope with this. I don't understand what's here. So some of these errors are fine. Some of them are just causing the natural variation that we see in the population. So I'm blonde, I got blue eyes, some of you have dark hair, some of you have brown eyes. And these are all due to sort of changes that the body can cope with. The body doesn't mind that there are some of this variation. But other times you can have changes that can make a huge problem. So how do we do this sort of really? So going about studying patients, and how have we done this in terms of the primary lymphedema cases that Saha presented. So if we start with the late onset, the lymphedema distichiasis, which was the one with the double row of eyelashes, this study came about by having lots of families, and you can see here in these pedigrees that a lot of the family members are affected by the disease, so they are marked with a black circle or black square. So you've got affected females and affected males here in these pedigrees. And the handle here, the clue here to put these families together in a study was that extra row of eyelashes, because we thought that must be something that makes this group unique. And therefore we had a very uniform group for studies, which is really important when you do genetic study. You have to have like really good uniform groups. And when we were then comparing the sequence of the patients up against the reference sequence, we then did find errors in the genetic code, and the errors were in a gene called FOXC2. So we can now start labeling not just the disease with a name, but we can actually also label it up with a genetic code, or a gene where we have found an error in the code. And as Saha said before, FOXC2 is really important for development of lymphatic valves. And as Chris also said, the lymphatics have these valves so that we make sure that the lymph fluid just goes one way. because we want it all to come out of the tissue and get circulated back into the system. So it's a little bit like on the tube where you go through a turnstile and they're trying to control you to go just in one direction. So if you've gone through the turnstile and you've decided that, oh no, I shouldn't go on the tube today, then you can't go out of the same one. You have to find another way out of the system. And this is how the valves work as well. They try to direct the flow in just one direction. And I have a little video here for you where you can actually see a lymphatic vessels that has been dissected out of a rat. So it's of course under an artificial situation, but it shows very clearly how the pulsation, so the muscles in the tissue, they get the vessel to pulsate, and that sort of opens the valve. And then when it relaxes, you can see that they are really tightly together, the two valves. So you can see over here how they are forming this really tight closure. So you can clearly imagine how the fluid will have problems getting back in again. So it's a very good system for directing the flow just in one direction. And with the FOXC2 patients, what we have found out now by looking at their lymphatic vessels under the microscope is that they are missing these valves, or if they have them, they are very misformed. They are just small lumps, and they definitely don't form this tight closure. And that explains then why you get this accumulation of fluid, because basically you have got nothing that can sort of help the fluid move up the body. So if we look at the lymph scans that Saha showed before, you can understand how you can get a really nice unidirectional tract in a healthy control in Peter. But in a lymphedema distichiasis patient where you don't have valves working properly, you can then understand now why you might get some fluid up through the system, but why it will also sort of move back down again, giving this effect on the imaging that we can do. So this is a very easy way to then identify these kind of patients when you do lymph scan imaging. So moving on to the other group of patients that Saha was presenting as well. This is the early onset, where you can already see the swelling at birth. And again, the study for Milroy disease was also based on large pedigrees. And again, you can appreciate, maybe this is not very clear, but there's loads of black squares and circles in these families. So again, we had some really nice large families that we could do genetic studies on. And again, we identified errors, but this time in a different gene. This time it was a gene called VEGFR3, which is a vascular endothelial cells growth factor. So it's a very important gene for the growth of lymphatic vessels. And so again, we can now start putting label on these disorders and also identifying the genetic mistake. But the problem we had here with Milroy, and as Saha also said, they don't have any other sort of clues really to make it a homogeneous group for genetic study. Only 70% of our congenital cases have mutations in VEGFR3. So we've been asking ourselves, what's happening in the rest of the patients? What's going on in the other ones? So by doing more work in the clinic, going back to case notes, really trying to tease things apart, we suddenly realized that actually there was a little group of patients here that all tend to have a small head circumference, so smaller than average head when you measure them with a measuring tape around the head. And we thought maybe that could be a helpful clue. And another thing that also became apparent when looking at the clinical note was that actually some of them also had problems with their eye. So they had, when you look at a photo of the retinal, so at a photo that's taken off the back of the eye, you can see that some of the retina is not very healthy. It's supposed to look like this up here. So there was actually two clues in this group of patients, and maybe that could help us to identify what's going on. So we did some sequencing, and we aligned it up to this reference genome, as I was telling you about. And indeed, we found that there was errors in their coding, and that all mapped to a gene that was called KIF11. So now we can put another label on another group of patients and give them counseling, explain to them what's going on, what is the risk of passing this on to future generations, and also if there's any specific things that we need to take care of in terms of management. And just to give you one more example on another group where just congenital primary lymphedema, but when again looking in detail in the clinical notes, we could see that there was a couple of families where there was a lot of miscarriages and a lot of fetal deaths, so children that die soon after being born because they had hydrops fetalis, which means water during fetal development. So it's basically swelling during fetal development. And also another thing was that a lot of the family members have atrial septal defect, which is a heart problem. So there was some good clues here as well that helped us then to identify that these two particular families had errors in the FB4 gene. So by doing these kind of studies, doing sequencing, aligning it up to the reference genome, we can start giving more and more of these patients an answer to why they have swellings and help them understand why they got this disease. But we haven't got the answer for all the patients here yet, so we're still working on it. And some of you might have heard about the 100,000 Genomes Project, which is a big initiative by the government to sequence 100,000 people in the UK, and again align that up to the reference genome and try and find out what's going on, and hopefully we can help more people understand what's going on and why they have whatever problem they have. And then the last couple of minutes of my part of the presentation, I'm just going to tell you a little bit about these two cases that Saha mentioned where they had somatic problems. So it was just part of the body that had this problem with a genetic defect. And when that happens, you got something called Mosaicism. So this is when only part of your body is affected by this genetic defect. So this arrow that we're looking for is not in every cell in the body. So we call that a somatic mutation or somatic variant. And it can happen at any time during fetal development. And the earlier it happens, the more cells will be affected by this. Because during fetal development, you got lots of cell divisions and you get more and more cells. And then in the end, you have a little human being that then grows up and becomes an adult. But if you have just one cell here that then get a mutation during the fetal development, because it can also happen during the fetal development, it can happen at any time. And if the body's DNA repair mechanisms haven't figured out that there's a little error that has been introduced in a sentence here, then it just goes on and gets passed on in the next cell divisions. So you can see here now there's, the cell has divided again, and now there's 3 cells with mutations in there. And it might be the cells that are forming sort of the buttock or the bottom part of the baby. So in a grown-up, you would then notice that there's something wrong in that section of the body. But this mutation could have been in one of the other cells that could have sort of been forming the basis of another part of the body. So depending on where this mutation takes place, it would be a different part of the body that gets affected. Mosaicism is very common in both in animals and plants, and it's so it's not an uncommon phenomenon. And again, like with some of the other variations I mentioned, like hair color and eye color, some of it is not problematic. So it's only a problem if you're a dog breeder and you only want golden retrievers because they fetch you more money when you're selling them. But actually, what's the matter with this one? That's pretty cute as well. So if you're not going to exhibitions with your pure pedigree, then I'm sure you could have just as much fun in the park throwing sticks with this one. But the reason why it's got black hair in some places and not golden all over is because it's carrying some of these somatic defects, but only in certain cells. So just back to our patient here that has this somatic overgrowth disorders. What we did to figure out, or actually this work was not done here at St. George's, but some collaborators of ours, they took DNA from some cells from this patient's upper body, and then they took some cells from the affected tissue down in her legs, and then they took and read the DNA, aligned it up to the reference sequence, and what they found was that in the PIK3CA gene, well actually let's start with this one down here, they found that there was a gene defect in these cells, but when they looked at the tissue from her upper body, they didn't find any errors in her DNA. So we now know that all the cells that are affected here has a mutation in this gene. And what we also know about this gene is that it's very important in a cell proliferation pathway. So it's a very important gene. And when it doesn't get controlled, when it's got a mutation, then it gets allowed to just proliferate and grow quickly, too quickly. So her legs are continuously growing. So you can Google her on the internet actually, and there's pictures from when she was little, and you can see her legs are not too bad, but as she grows older and becomes a teenager, they get bigger, and now here you can see how enormous they are. The good thing about some of the mutations that we find is that they are in genes where we already have some way of controlling, because there's been a medication developed for this particular pathway that the gene fits into. And therefore, there is a hope now that maybe by putting this patient on this kind of medication, that we can stop this growth of her cells, so that maybe her legs are not going to get any larger than they are. And then I'm going to hand over to Peter to talk a little bit more about treatment.
Now, I'll try not to keep you too long from those huge muffins that are at the back. And I'm going to talk about the clinic and treatment. If I can wind myself up. And at St. George's, we have the biggest clinic, lymphedema clinic in the UK. We see more patients here than anywhere else. And this helps us because it's the patients then that pose the questions to us scientifically. What's wrong? What can we do to find out? How can science answer this? And then hopefully when we've got some answers, we go back to the patient, explain it, and then see if we can improve the treatment. Now, which is forward. We treat all forms of lymphedema here. You've heard of secondary lymphedema, where there's some external cause, such as removal of a lymph gland in cancer that causes lymphedema. And in fact, if you mention lymphedema in the street to most people, they'll say, isn't that caused by cancer or cancer treatment? And the answer is in part yes, but actually breast cancer-related lymphedema, where you get a swollen arm from removal of cancerous cells under the arm, makes up only about 1/8 of all forms of lymphedema. There are lots of different forms of lymphedema, and as Christiana mentioned, see it that side as well. You mentioned elephantiasis. That's not the elephant man, that's a different genetic disorder. Elephantiasis is having such a swollen leg, it resembles an elephant's leg. And that's why it's called elephantiasis. But this too is a form of lymphedema. It's probably the commonest type in the world and is caused by a mosquito bite, which is why you tend to see it in tropical countries. Now, our talk tonight is concentrated on primary lymphedema and the genetics of primary lymphedema. But I believe that even these secondary forms, so you get lymphedema after cancer or you get it from a mosquito bite, I bet you eventually we'll find out there's going to be a genetic predisposition to getting it, because not everybody who has cancer treatment develops lymphedema, and not everybody that gets a mosquito bite in India gets elephantiasis. So what about treatment? Well, one of the problems with lymphedema is because there's been so little research into the science of it, the biology, and understanding it, our treatments are poor. And you'll see over here how we treat it, mainly with things like compression garments and exercise and massage, and I'll come back to that. There are ways of trying to treat lymphedema, such as super microsurgery, but there's not one operation as yet that fits all types of lymphedema, and surgery is still in the early stages of development to try and make it work for lymphedema, so it's not generally a treatment that's available. Drug treatment, because let's face it, when you go and see the doctor, what are you usually offered when you've got a disease? You're offered a drug or an operation, generally speaking. You might be sent off to the physiotherapist and have some other treatment, but in general, we're a drug and a surgery-based medical community. And the embarrassment is that there is not one drug yet developed to make the lymph system work better, which is a real embarrassment. If you've got a heart problem or a problem with the heart and circulation, there are hundreds of drugs that work on the heart, but not one drug that makes the lymph system work better. So this is the challenge, and it's only through science that we're going to make improvements in that regard. Now, so what do we do then if we haven't got a drug and we haven't got an operation? Well, what we do is we try and make the lymph system work as well as it can work. So let's say someone's got a swollen leg from primary lymphedema. What do we do? Well, under normal circumstances, what makes the lymph system work is movement and exercise. The heart pumps the blood round, but your lymph fluid only moves if you move it. So at bed at night, it actually falls to a trickle. But as soon as you stand up and walk around, your lymph system responds very briskly and quickly. So hopefully one doesn't get any degree of swelling. Having said that, lymphedema is considered rare, but it's probably not that rare. Anyone here gets a swollen ankle in the heat or on a long-haul flight? Well, that is because you haven't got lymphedema, but that's because your lymph system is not working well enough. And that's how all lymphedema starts, usually with a bit of swelling, which then is allowed to progress. Fortunately, most patients, or most people, that swelling won't become a problem. But as you get older and your lymph system gets weaker, then it can become more of a problem. So any ankle swelling reflects that the lymph system not working very well. So what do we do to make it work better? Well, we encourage exercise and movement, even with someone with a lymph system in one leg that's not working very well. But how do we enhance the effect of the exercise and movement? And that's where the compression garments come in. Because if you then compress the part, you change the pressures to allow fluid to drain to the lymph glands that much better. It may seem an archaic system, but it's simple, it's safe. You're applying basic physiological principles, may not be state-of-the-art science, but that's how we treat patients. And if we've got someone with a very big leg, you can't just put a stocking on and tell them to walk around. You have to actually get the swelling down. So someone like that poor chap in India, we would then use an intensive period of treatment to try and get the swelling down, and then try and keep the lymphedema under control. So we can't cure it, we try and control it. And if you control the swelling, then they can walk better, they don't get infections quite so much, and life's an awful lot better. So the intensive treatment involves using multiple layers of bandages applied in a very skilled way. It's not just throwing or putting any old bandage on, and our therapists are very skilled at doing this, and they'll probably do some massage as well to try and further increase lymph drainage, because massage also moves the lymph onward. So this is protecting the skin. We have to even treat the toes, because the toes become very swollen, and if you just put a bandage on around the ankle or the leg, you'll make the toes worse. So we have to treat the toes or the fingers if it happens to be the upper limb. We then have to put a lot of padding on, because then when we put the compression bandage on top, it doesn't cause sort of sores and it doesn't press on any particular part of that limb. And then the compression bandage is quite rigid, so that they're walking away, and the pumping effect of the muscles within is what actually gets a lazy lymph system to work better. And then once we've got the swelling down, then we can apply a compression garment on an arm or a leg. This is more difficult if you happen to be male and have swelling of your privates, or have swelling of your face, or some breast cancer patients have lymphedema and swelling of the breast. That's always more difficult to treat. But most lymphedemas affect the limbs, and that's why we use the compression garments. But the exercising is still absolutely critical. And of course, compression garments aren't fashionable. They're not sexy. Anyone as old as me will remember a comedy program called Last of the Summer Wine. Jenny, you're not old enough. We have our principal and boss of the university. It's lovely to see her here, but she wouldn't be old enough possibly to remember Last of the Summer Wine. But there was a character in it called Nora Batty, and she used to wear stockings that were always wrinkled and falling down. And one of her neighbours was a lustful old man who just couldn't get enough of these stockings falling down. So when patients come to see me, they often say, "Oh, I'm not going to wear those awful Nora Batty stockings." But they needn't be terrible. They've made them much more elegant, although to be effective, they still need to be quite strong and rigid. But I put a picture of Serena Williams to just show how garments can become more fashionable, because in athletics now they've become much more fashionable to wear compression garments. The Brownlee brothers and their triathlons wear them, and Serena Williams was wearing them at the US Open. And what's that doing? That's actually improving lymph drainage, so that muscle recovery is that much quicker, because if you get the lymph drainage, you're clearing out the toxins, the waste, the sewage, everything from the muscle, and the muscle works better. Of course, the athletes don't know that it's actually the lymph system, but it just goes to show if you make something fashionable, even the top athletes will start wearing it. So the principles of understanding the lymph system and how you make it work better are very important. And what has been our limiting factor over the years has been a lack of knowledge of the lymph system. And our lack of knowledge scientifically is because we haven't really had the tools to fully understand what our lymph system does, what happens when it goes wrong, and that's why it's very much a Cinderella system compared to the heart and the circulation. Everybody knows about that, whereas nobody knows about the lymph system. If you go and see your GP and ask about the lymph system or lymphedema, you'll probably end up with a blank face, because we're trying to get it taught better in medical schools now, like St George's, so that all the doctors understand the lymph system. But this quote I like, because it just shows you that if you've got the techniques, such as the genetic techniques that are coming through now, you can learn a lot more, and then the interest follows, and then hopefully better treatments will follow. So the scientific understanding of the lymph system is critical, and that's what we're trying to do here at St George's, is to understand it better through the genetic basis, even eventually to understand the secondary lymphodemas. And this is not just for self-promotion, but you cannot do research without money. And we've been very fortunate here at St George's to have excellent funding from the British Heart Foundation from New Life and also from the Medical Research Council, which is currently funding us. And that's enabled us to make the discoveries that we have done, and hopefully we'll make more in the future. So at that point I'll stop and hand back to Julian, who will tell you what happens next.